Students, in this video we are going to find some of the questions which has been asked in the previous year's exams from the chapter Biotechnology, Principles and Processes. In gel electrophoresis, the separated DNA fragments can be visualized with the help of ethidium bromide in UV radiation. BT cotton variety that was developed by introduction of toxic gene of Bacillus thuringiensis is resistant to insect pest. Identify the wrong statement with regards to restriction endonuclease enzyme. They can cut the DNA strand at palindromic site. This is the right statement. They are useful in genetic engineering. This is also right statement. Sticky ends can be joined by using DNA ligase. This is also right statement. The wrong statement is each restriction enzyme function by inspecting the length of DNA sequence. This is a wrong statement because exonuclease cannot inspect the length of DNA sequence. So this is a wrong statement. The palindromic sequence which is recognized by EcoR1 restriction enzyme is GAATC, GAATTC. This is the right statement. The sequence that controls the copy number of the linked DNA in the vector is termed as origin of replication site, ORI site. The selectable markers is used to eliminate non-transformants. Non-transformants also called as non-recombinants, so that transformants, recombinants can be regenerated. Given below the four statements pertaining to the separation of DNA fragments using gel electrophoresis. Identify the incorrect statement. The incorrect statements are DNA is negatively charged, right? And so it is loaded on gel towards the anode terminal. No. So as the DNA are negatively charged, they are going to loaded at the negative terminal so that they can migrate towards anode. DNA fragment travel along the surface of the gel whose concentration does not affect the movement of DNA. No, the concentration of agarose gel definitely affects the movement of DNA. This is also the incorrect statement. Then pure DNA can be visualized directly by exposing UV radiation. No, we need to use ethidium bromide also. So this is the wrong statement. So the right statement is smaller the size of the DNA fragment, larger is the distance it travels through. So first statement, second statement and fourth statements are incorrect statement for gel electrophoresis. An enzyme catalyzing the removal of nucleotide from the ends of the DNA is exonuclease. From the ends of the DNA, exonuclease, at a specific site, endonuclease. Match the following enzymes with their function. Restriction endonuclease enzyme, they cut the DNA at a specific position. Restriction exonuclease enzyme, they are going to remove the nucleotides from the ends of the DNA. DNA ligase enzyme joins the DNA fragments and tag DNA polymerase extends primer on genomic template. So option C is the right option. The two antibiotic resistance gene on the vector PBR322 is ampicillin resistance gene and tetracycline resistance gene. Which one of the following equipment is essentially required for growing microbes on a large scale in industrial production of enzyme? It is bioreactor or fermenter. Following statement describe the characteristics of enzyme restriction endonuclease. Identify the incorrect statement. The enzyme cut the DNA molecule at identified position within the DNA. This is the right statement for endonuclease. The enzyme bind DNA at a specific site and cut only one of the two strands. No, this is the incorrect statement regarding endonuclease. Which of the following is commonly used vector introducing a DNA fragments in human lymphocyte? It is retrovirus. Disarmed retrovirus is used. The correct order of PCR, polymerase chain reaction, first step is denaturation, second aniline and third is extension. The DNA fragment separated on agarose gel can be visualized after staining with ethidium bromide and exposure to UV light. The gene whose expression helps to identify transformed cells recombinants is selectable markers. What is the criteria for DNA fragments movement on agarose gel during electrophoresis? Gel electrophoresis. The criteria is smaller DNA fragment size, further it's moves. Smaller the DNA fragments, they can move faster in the agarose gel. Which of the following is not the feature of plasmid? Plasmid is single-stranded is the wrong statement. Plasmid is double-stranded, independently replicated, circular in structure and transferable. These are the right statements. Tag DNA polymerase enzyme used in PCR is obtained from Thermus aquaticus bacteria. Which of the following is a restriction endonuclease? It is HIND2. The cutting of DNA at a specific location becomes possible with the discovery of restriction enzymes. Which of the following restriction enzymes produce blunt end EcoRV? Which of the following is not component of downstream processing? Downstream processing, not a part of the downstream processing is pure expression, whereas preservation, purification, separation are the parts of downstream processing process. A single-stranded nucleic acid tagged with a radioactive molecule is called as probe. It is used in DNA fingerprinting. There is a restriction endonuclease called EcoR1. What does COPE part stands for? COPE part stands for coli. Agarose extracted from seaweeds find used in gel electrophoresis. So these are some of the questions of this chapter which has been asked in previous years of NEET exam. Thank you, students.